Hello and welcome to Nikolay's genetics lessons, and in this video we are going to talk about protein folding. And here is a problem: which of the following peptides is more likely to take an alpha helical structure and why? And here is the first sequence and here is the second sequence. These two pictures of the proteins has nothing to do with our problem and the sequences. I just want to use them to demonstrate the beauty of the proteins and also the complexity of the structures. But if you take a closer look, you will find that basically we can see here only three structures, which we call secondary structures. First would be alpha helix. So here's the alpha helix here, and in many places you can see in this protein, and also beta sheets, or we also call them beta pleated sheets. And also you can find here irregular structures like this. So we just don't have special name for them because they're too complex to describe, so we just call them irregular structures. And all these three secondary structures make about 33% each. In some proteins we can see that one type of structure would be overrepresented, in other different. But on the overall, if we consider for example 1000 proteins, we will find that alpha helix, beta sheet and irregular structures would make about 33% each. Now back to alpha helixes. If we consider this sequence and this sequence, you actually even don't have to know all this one letter abbreviation of each amino acid. You just have to know at least two amino acids in the abbreviation. First would be glycine, and G stands for the glycine, and we have here one glycine, another glycine here, and here's the third glycine. And you cannot find glycine in this sequence because glycine is the most flexible amino acid, and its flexibility just disrupts alpha helix structure. And those rarely, but we can find glycine in alpha helix, but one amino acid, which is proline, we almost never can find in the middle of the alpha helixes because its rigid structure, it is most rigid amino acid, just disrupt alpha helix. And now, as you see, the first sequence of amino acid doesn't have no glycine, no proline. So this is going to be our choice. This amino acid sequence is more likely to make alpha helix. And this is all for today. Subscribe and see you in the next video. Goodbye.